Good morning, Science 10. I hope that you are all having a nice start to your Thursday and you're enjoying your last day of isolation. This is going to be our last video lesson. So just some housekeeping. We have the lesson 1 to 3 practice problems are overdue now. If you have not taken the chance to complete those, please do. You will find all the answers to that in video lessons 1 to 3, okay? Today I'm going to post practice problems associated with lessons 4 and 5, okay? I will make those due tomorrow, okay? So that should give you time to kind of watch the videos, answer those lessons, and then that leaves you lots of time to study for the physics unit test on Tuesday, okay? And remember, we will have a unit review for physics on the Monday as well, okay? But that does not mean that you can not study for physics for the weekend, okay? It's important that we are brushing up and kind of revisiting that often so it's kind of deep in our brain, right? All right, so let's get going with the remainder of the biology topic. Okay, so we're going to start talking with just a little refresher on the cell membrane. Okay, this is really important. The cell membrane, remember that it's made up of mostly 2 components. So it's proteins and the phospholipid bilayer are the 2 main components of the cell membrane. Remember the phospholipid bilayer, it has, it's bilayer because there's 2 layers of it. So we have the phosphate water-loving head pointing to the outside, and we have the hydrophobic lipid tails pointing to the inside, okay? And then we have the proteins, which are embedded in the phospholipid bilayer, and these are involved with some forms of cellular transport, which we're going to talk about today. So again, the cell membrane is a semi-permeable membrane because it selects which substances can get into or out of a cell, okay? And those substances are going to have unique chemical and physical properties. Usually we have receptors on those proteins that are in the phospholipid bilayer. Okay, there are going to be receptors there which are unique to these substances, and that's how the cell is semi-permeable, because the proteins on the surface will recognize what the substances are, and it pretty much gives the go-ahead whether or not it can be let into a cell or not. Okay, yesterday we talked about diffusion. So in general, this is the tendency of particles to go from an area of high concentration to an area of low concentration. Okay, think of making iced tea as a good example for this. A concentration gradient is when we have a difference in concentration gradient, a difference in concentration across a membrane. Okay, so when there is a different amount of particles on the inside or outside of the cell, kind of like what we have in this image on the bottom right-hand corner of your screen, we would say that there is a concentration gradient because there's more particles on the outside of the cell there. And again, those are regulated by protein molecules that are in the phospholipid bilayer to make sure that the internal cellular conditions are ideal for that cell. Okay. These are just 5 kind of points on concentration gradient. So it will drive diffusion of osmosis. It must involve different concentrations. In cells, the different concentrations may be separated by a membrane. Okay, and this is not exclusive just to cells. We're going to talk about reverse osmosis water treatments today. Okay, and this is how water is kind of filtered using technologies. Okay. So this slide I had to fix from last day. So osmosis, this is the diffusion of water across the cell membrane. And this occurs when the solute cannot pass through the membrane, but water molecules can. And they will move against their concentration gradient in attempts to kind of reduce the difference in concentration within that system itself. So in here, we'll have a net movement of water to the right. Okay, so hypertonic is when there's more solute outside of the cell. So tying into the osmosis slide that we just talked about, if there's more solute concentration outside of the cell, the water is going to leave the cell in order to kind of reduce that concentration difference. Isotonic, if there is no concentration gradient, the water is not going to move. And hypotonic, if we have more solute inside of the cell, the water is going to move into the cell to lower the concentration gradient that we experience inside and outside of the cell. Okay, make sure you know these. Okay, make sure you know hypertonic, isotonic, and hypotonic. They will be quizzed on assessments. Okay, this was an animation that kind of showed how osmosis works. I'm not going to play that again. I did not get in trouble for that, and my video is still up on YouTube, so that's excellent. Okay, so this is new content today. So the movement of water, so particularly in cell plants, or this is only found in cell plants. So the term is called Turgor pressure, and this is the movement of water into the central vacuole of the plant cell. Okay, so remember the vacuole is kind of like a storage unit for plant cells. Okay, vacuoles are only found in plant cells. Okay. Now, when the plant has lots of water, like in the soil, that vacuole will be full and expanded. So this is going to actually help give the plant some of its structure by having a filled central vacuole. When there's not as much water in the environment, that vacuole is going to shrink because that water is going to be transported to other parts of the plant that it might require that water. So when that vacuole shrinks, the plant kind of looks wilted. And this on the right side of your screen is a piece of celery, which looks like it is kind of wilted and dying, right? But if you were to put that empty or that dying piece of celery into a cup full of water, it will take up the water that's in the cup and it will fill up its central vacuoles. So if you ever have any celery at home that is uneaten and it's kind of wilted, you can return it back to how it was purchased from the store. If you put it in a cup of water and wait a couple days, that water will eventually make its way up into the stalk, into the leaves of the celery plant. All right. So now we're getting into the regulation of transport in cells. Okay, so remember the cell membrane is really important in what regulates what can go into or out of a cell. There are some particular chemicals and molecules that are really important, and they do not need to be regulated by a protein, okay? So we're talking about passive transport right now. This type of transport does not require any energy input for it to occur. So if we look at this image on the right here, we have a concentration gradient because there's more chemicals outside of the cell than inside of the cell. And because these molecules are small enough that they are actually able to diffuse right through the cell membrane. Okay, they're not really large, so they don't need a protein channel. They can just kind of like sneak through the side gate. Okay, and usually these molecules that can diffuse are really important for a cell. So some examples of chemicals that enter the cell through diffusion are water molecules, oxygen, and carbon dioxide. Okay, so we require oxygen for cellular respiration, so we're able to like move around and stuff like that. So we need oxygen to be able to quickly come into a cell. And carbon dioxide is a waste product of our respiration. So again, we can't have that carbon dioxide accumulating within the cell because it could be toxic for it. So it's important that that is also able to readily diffuse out of a cell. Okay, so that is passive transport. A big takeaway for that is no energy is required for that to occur. It's passive. Okay. Next, we have facilitated diffusion. So this is where we need a protein channel to get through the cell membrane. So proteins embedded in the membrane will allow these particles to move across the membrane. And this movement is in response to the concentration gradient. So if we look up here, we have a higher concentration of solutes outside of the cell. So naturally, things want to eliminate that concentration gradient. So in facilitated diffusion, that concentration gradient is reduced by protein channels, which will allow these solutes to move across the cell membrane. Okay, an example of this is sugar in your bloodstream. So sugars, once it's in your blood, it's going to need a protein channel to get into your cells, which can then be broken down and then used for energy. And that is done in the mitochondria. Remember, the mitochondria is the powerhouse of the cell. Okay. Next, we have active transport. So this is where we have particles are being moved against their concentration gradient. Okay. So we need carrier proteins to move these solutes, and it requires energy to move things against their concentration gradient. So think of your iced tea example again. So to diffuse it and make it a homogeneous solution in your cup, it doesn't require much energy. Like if you were just to leave the crystals in the bottom of the cup without stirring, eventually the solution would be homogeneous. Okay. But if we wanted to kind of like filter the iced tea out of the water, so to make it go from an area of equal concentration to high concentration, we're going to have to put energy into that. Same kind of idea here. We need energy to move things against their concentration gradient. So we've talked about the sodium-potassium pump. This is really important for our cells. Okay, so cytoplasm is inside of the cell. It has a low amount of sodium and a high amount of potassium. Okay, it wants to keep the amount of sodium low because it could interfere with cellular signaling. Okay, so it will pump sodium outside of the cell where there's already a high concentration of sodium, a higher concentration when compared to the inside of the cell. So we need to put in energy in the form of ATP to move these solutes across their concentration gradient. Okay, ATP is the usable energy that our cells use for cellular functions, and that is created in the mitochondria. Okay, so transportation in cells. Sometimes the molecules are either too big. Sometimes they're just too big to move through channel proteins that are in the plasma membrane. So a strategy to kind of get around this is that cells can create vesicles to bring large objects into or out of a cell. Okay, so this top diagram here, this is an amoeba feeding by endocytosis. So there's particles outside of the cell. It creates a vesicle to kind of bring those particles inside the cell, which it can then digest and acquire the energy from. Conversely, we have exocytosis. So this is where we are eliminating something from the cell. So exo is outside, endo is inside. Okay, in this case, the vesicle is still going to fuse with the with the cell membrane, and it is going to release and get rid of its contents. Okay, so this is how some sorts of waste products are removed by the cell, is by exocytosis. Okay, so just kind of talking a little bit more about endocytosis. So the vesicle is going to form around the cell membrane and kind of pinch around it to create that vesicle to bring it inside of the cell. This is how some organisms feed, and this is also how some viruses gain entry into the cell. Okay, so amoebas will ingest their food this way. Okay, and we're going to show a little clip about an amoeba eating paramecium. So this guy here is the amoeba, and this little thing is a paramecium. So we can see the amoeba kind of forming that vesicle around the paramecium. If we look down here, this is cool. This is a 400 times magnification of an amoeba. And now we can kind of see the amoeba is starting to pinch off that vesicle. And this paramecium is not looking like it's very happy. It's scared. It's giving its last words right now. Goodbye, cruel world. And so that is how an amoeba will eat via endocytosis. Okay, now exocytosis. This process is used when cells release large waste products or when secretory cells, so like when cells will, secretory cells are those that will like release hormones that will travel throughout the bloodstream into target cells in your body. Okay, when they release signaling molecules like insulin, for example, they will be released via exocytosis. So remember, exo is getting rid of something. Okay, so some signaling molecules are neurotransmitters. So like acetylcholine would be one, epinephrine, stress hormones. Okay, these are some sort of signaling molecules, and that's how they're released into the body. So remember, the endoplasmic reticulum is where proteins are being created. In particular, the rough endoplasmic reticulum, this is where the ribosomes are located. And remember, the ribosomes are the sites of protein synthesis. Okay, so protein hormones made in the ER, will be secreted into the bloodstream via exocytosis. So an example of this is insulin. Insulin is produced by the pancreas. This is involved with regulating our blood sugar. Okay, so after a meal, our blood sugar increases. So insulin will be produced by the pancreas and released via exocytosis. It will travel throughout the bloodstream until it binds with a protein on a plasma membrane of a cell. This will signal to the protein to allow the glucose to be taken into the cell through facilitated diffusion. Okay, so that will not take energy for the glucose to go into the cell, but it does require a protein channel for it to be transported into the cell. Okay. So considering what we have talked about today in terms of cellular transport, scientists use these concepts of cellular transport to kind of help solve some problems that are faced by society. So HIV, human immunodeficiency virus, this will target protein receptors on the immune cells. So if we look at our this image on the right side of your screen here, We have a protein receptor in the cell membrane here. And here's kind of like a simplified image of a virus. Okay. Viruses will have a particular binding site, which will interact with a binding site of a receptor protein. It's kind of like a lock-in key kind of idea. Scientists who are trying to reduce the effects of HIV have developed a kind of like a receptor protein blocker, and this prevents the association between the active site on the virus and the active site on the receptor protein. So if these two are not able to bind, the virus will not be able to inject its genetic material into the cell, which would further destroying immune cells, especially in the case for HIV. Cancer treatments, some cancer treatments will attempt to block the receptors so the virus cannot bind to the immune cells and then inject their genetic material. Okay, just some more applications. So liposomes, these are fluid-filled sacs surrounded by a phospholipid bilayer. So these can be synthetically made. So synthetic is you're not going to be able to create it in nature, but humans are able to produce it in a laboratory under particular conditions. Okay, so they can make these liposomes, and then they can like put in chemicals or hormones or synthetic insulin, whatever the person needs, into these liposomes. And then these liposomes will then transport throughout the body, fuse to the cell membrane, and then deliver the materials, whatever is required by the person. Okay. So for some people who are diabetic, this could be a strategy how they deliver insulin into the body. Yeah, so diabetics have a resistance to the blood sugar. So they have to treat their blood sugar levels with synthetic means. Okay, so and we're going to talk about dialysis. So dialysis, this is a treatment for dysfunctional kidneys. So kidneys are essential to filter our blood. Okay. Yeah, to filter our blood to produce urine and waste products. Okay. Dialysis is based on the principles of diffusion, osmosis, and the operation of concentration gradients. And hemodialysis, this is one form of dialysis. This is where the blood is going to be removed from the body and cleaned using a machine. Typically, this is done at a hospital because these hemodialysis machines are quite expensive, although I know that some people are able to do this at home. Okay, another type of dialysis is peritoneal dialysis. So this is where fluid, so the dialysate, is filled. It has a concentration of electrolytes and sugars. This is going to be inserted into the peritoneal cavity, which lines the stomach. So because in the blood, there's going to be a higher concentration of toxin and waste materials, it will diffuse into the dialysate because there is a lower concentration there. So remember, diffusion, it always wants to go from a high concentration to a low concentration. Okay, so over time, toxic and waste materials will move into the dialysate. And then when the dialysate becomes saturated, it is removed and replaced with another dialysate bag until the process is complete until all the toxins and waste materials are removed from a person. Okay, desalination. This is really important for a society as we go forward. We know that about 78% of the Earth's surface is covered in water. Unfortunately, the majority of that is salt water, and only about 1% of all the water found on Earth is fresh water. So we don't have a water problem, we have a salt problem, okay? So desalination is tricky because it's really expensive to do on a large scale, but it's really important and necessary for our society as we're trying to provide food and water and internet, you know, like basic resources for all people on the earth. So desalination is going to be really important for the future. So if anybody's looking for potential careers or any breakthroughs like that, maybe desalination practices would be a good career to get into. Okay, so desalination, definition of desalination is removing salt from seawater or just water in general to make it suitable for drinking. We can desalinate water through a process called reverse osmosis. So this is where water moves from a low water concentration, so lots of salt, to a high water concentration using pressure created by a pump. Okay, so this image right here, we have regular osmosis. So solvent enters the solution along the concentration gradient. So because there's more salt over here, the water is going to want to move to the side that has more salt. So it reduces that concentration gradient there. However, in reverse osmosis, they use pressure and a pump to move the water against the direction that it normally would. And that is how solute, so as that water is pushed across that membrane, the solute will, the membrane is semi-permeable, impermeable to the solutes, but permeable to the water. So the water is able to get through, but the solutes will not. They'll get stuck in that filter, and then we'll be left with desalinated water on the right, okay? Reverse osmosis, this is done, and reverse osmosis is a strategy to desalinate water, but we do need to apply like energy to desalinate that water. So here is a video of reverse osmosis.
The core technology used at the Adelaide desalination plant is called reverse osmosis. First, let's explain what osmosis is. Osmosis is a naturally occurring process in which a liquid, such as water, spontaneously passes through a membrane. The membrane or semi-permeable barrier allows some molecules, like water, through, but other molecules, like the majority of salts, are unable to easily pass through the membrane structure. The flow of liquid through such a membrane occurs naturally to try and even out the salt concentrations between the two solutions. That is, the liquid flows from the less concentrated solution, such as fresh water, to a more concentrated solution, such as seawater. When the direction of liquid flow is reversed, it's called reverse osmosis. By pressurizing the concentrated solution, in this case seawater, we are able to force water molecules in the reverse direction, from the salty seawater side through the membrane to the freshwater side.
Excellent. So I hope that was a good little summary there. And yeah, so that will be the last of today's lesson. So I will post this to the Google Classroom, And then once you have finished watching this video as well as the remaining, the other videos, please take the time to complete videos 3 to 5 practice problems, which are going to be due for tomorrow. Okay. So a reminder, we're going to have a physics unit test on Tuesday of next week. And then we're also going to have a biology chapter 1 and 2 quiz of Thursday of next week. Okay. So we have lots of assessments next week. So make sure you take some time this weekend to get some studying done. Okay. As always, if you need help with anything, feel free to send me a text or an email or give me a phone call. That's totally fine with me. I'm here to help you guys. And yeah, thank you for your attention and your time during these last couple of weeks learning online. And I look forward to seeing you all in person next week. All right. Take care and enjoy your weekend, guys.